Good evening. What a pleasure it is to come to this event, and a great pleasure to be able to introduce our speaker, Professor Scott Selleck. One of the best things I did as dean was I encouraged Scott to leave the University of Minnesota and come to Penn State. And the institution was better from the start. In the beginning, Scott was head of the Department of Biochemistry and Molecular Biology, and he did a wonderful job there. A lot of it has to do with supporting the faculty in the department, making sure that courses are being taught appropriately, and recruiting new faculty members. And Scott and I together did some work on recruiting, and we had good success. We got some really good people. And he was in that capacity for many years. I forget how many years, Scott. 8 years, yeah. And he left the department better than it was when he started, but because he was, as I say, the thing I mostly did with him was to recruit new faculty members, but I also supported some of the other elements of the department. So some years ago now, when Scott stepped down from serving the department to concentrate on his research, which has been terrific, and on teaching, where he focuses not only on the science, but also on what the students need to do to be successful. I think I know Scott from my friendship and a relationship with him, but when I hear him talk about his teaching, I think, one, it's great, and two, there's a little bit of pressure on the students to do the right things. And I think that's great. It's a teacher who is a fantastic scientist and teaches the science, but also urges the students to be doing the things that'll make them successful. Scott's research is not just exciting science, but it also has the potential to be transformational in treating some serious diseases. Alzheimer's is the one we talk about the most, but I think there are related diseases. Scott, I'm delighted to be here and to have the privilege of introducing you and learning more about your exciting research.
Well, good evening, everyone. Thanks for coming. Thanks for supporting our institution and our college. And thank you, Dan, for recruiting me to Penn State and giving me opportunities to do things. And so there are many people I want to thank. I want to thank Mike and Kelly, who's done so much to make this evening possible. Tamara Morris is here from the Office of Technology Transfer, and Tammy is helping us get this technology to drug development and into people. So there are many people to thank. And so this evening I want to tell you something about neurodegenerative disease, where we are, something about its history, and what are the prospects for the future. And those include some of the work that I'm going to tell you a bit that we're doing. But before I jump into that story, I want to say a few things about our college. So it's important, so I'm just one representative of this college, right? I'm one of hundreds of faculty in 7 departments: mathematics, statistics, physics, astronomy, chemistry, biochemistry, biology. And I am not kidding or being deceptive or overrating when I say this college is world class. And let me give you some data to support that. So when Dan was the dean, the National Academy of Science actually did assessments of science programs all across our country. And they actually had metrics. They weren't just opinions. So the U.S. News and World Report, by the way, that's opinions. Opinions of people that are in the fields, et cetera, et cetera. But it's opinions, inherently divergent. The National Academy said, we want real data. So they had, I think, 22 different metrics for which they evaluated all these different science programs across the country. And when Dan saw that data, he said, wow, these numbers are, on chemistry departments and biochemistry departments and astronomy departments. But if we take all those numbers and we consolidate them, they can represent the college, not just an individual department. So Dan took those numbers and did all the calculations for all the major public and private universities in the United States. And I think it's safe to say that even those of us, and Dan in particular, who knew that our college was really good, the numbers astonished him. So Penn State was in the top 10 colleges of science in the country, of all colleges. And so, you go through that list and you go, okay, well, where are we compared to, no offense intended, to Duke or something? And what we learned from that was people's perceptions were not in unison with reality. Those metrics express reality. And of course, we were delighted because it told us something that we knew in our bones, but we didn't have the data. So when I tell you that the College of Science, then and now, is a world-leading college, we have data to back it up. So I wanted to make that, and it hasn't changed. The College of Science, its revenue in research dollars continues to go up, despite our rather uncertain times. Okay, we're still recruiting actively. So the College of Science is sound and healthy, despite the challenges that we face. So this is the topic for what I'm going to tell you about, the history and biology of Alzheimer's disease, but I'm going to tell you about other disorders as well. And obviously, research is a very important domain of what we do, but it's not the only domain. We are responsible for the education of our students. And I have come to learn that we shouldn't just teach them about the facts and figures and principles of our disciplines. We have to give them some idea of their place. In other words, what are their motivations? What are the motivations and values of someone who takes on a career in science? And in some ways, it's what motivates you through the difficult times, when your experiments aren't working, when a beautiful idea is shot down by an ugly fact. And so it's important, I've learned that in talking to our undergraduates and our first-year graduate students, that there's value in talking about the best way I can put it is the motivations and values of being a scientist. And I just want to spend a few moments on what, and of course those are values that I think about every day. So what is it that motivates transformative science? This is a picture from one of the leading architects in the United States. He designed the Chicago Columbian Expedition that was in 1893. He transformed American cities. He designed Cleveland. He designed the Union Station, right? So this gentleman really changed the way not only Chicago was laid out, but Cleveland and other cities. He really was transformative in how we thought about architecture. But he's probably, in addition for being known for that, this gentleman was known, his name is Daniel Burnham. He's known for this famous quote: "Make no little plans. They have no magic to stir the blood, and probably themselves will not be realized." Make big plans. Aim high in hope and work. Oh, I love that last part. Your work ethic has to match your hope and your aspirations. It ain't going to come easy. You can hope all you want, but if you're not willing to get in there and work, it is not going to happen. So as I sort of rephrase this to some of my students, and I say, well, the status quo is not inevitable. It can be changed. And what am I really telling them? I'm telling them, you are the agent of that change. It's not going to happen if you as an individual decide that it can't be changed. If you're in a state of hopelessness, the world is messed up, we got all kinds of problems. If you don't decide that you can move it, you won't try. So, status quo is not inevitable. Put another way, what has motivated human beings to try and do great things? It's the power of possibility. What would the world be like if this idea that I have, this could be transformed into reality? People that do great things have to bear this in mind, and because it would help them get over those times when things aren't working. And so I've come to incorporate these, I hope to be words of encouragement to our students. And basically the message to them is really simple. You make a difference. You are not a number. You are not just one applicant to this medical school, to that graduate school. You have value. You must remember this, because without that I don't mean arrogance, I mean value, that it motivates them to try. And it's critical for the faculty in our college. If they don't believe they can do something, they're not going to try it. So I think it's fundamental to what we do in science, to having the aspirations and the humility to try, to hope big, and to work hard. Okay, so that's my 4 minutes of philosophy So let me tell you about some of the powers of possibilities that have played a role in my life. And I have to acknowledge my mother for many reasons. Number one, she was the smartest person I've ever met in my life. Sorry, Dan. And she died of a neurodegenerative disease called Lewy body dementia. And I'm a physician. I did an MD and a PhD together. I've taken care of Alzheimer's patients and Parkinson's patients, but when it's your mom, it's different. First of all, you know them. So you see all the changes that occur. So my mom died of this disorder in 2010. And like I said, she was the smartest person I've ever known. This is her stomping around in Egypt. And this is my mother hanging out with my brother and I when I was much shorter but a lot cuter in Milan. And this photograph was with my brother a little bit later. And I watched her go through these disease processes. And at the time I first assembled some of this, the number of people living with Alzheimer's in the United States was 6 million. It's now, that was only 2 or 3 years ago. It's now 7.2 million. And we're making progress for other forms of disease, like heart disease. It's going down. Our treatments are working. Not for this class of diseases. So there's work to be done. And I watched my mother's demise. After her diagnosis, she was dead in 4 years, which is not an atypical course. She had a combination of Parkinson's symptoms and Alzheimer's. So she had movement disorders and memory disorders. And I have to tell you, she had a swallowing deficit. And about 4 years after her death, a PhD student in my lab made some observations that said we had the possibility of pushing back on these diseases at a very root mechanistic level. I was thinking about retiring. I just finished being a department head. I was going, hey, I can ride my electric bike across Italy, no problem. But I said, I can't do it. There was an observation there that I could not let go. I'm so glad I didn't. So, her life and her example are certainly part of my what is possible. So let me tell you a little bit about these disorders, put them in context for you. First of all, there are no cures for these disorders, okay? Tragically, they're not out there. And I've given you 3 major ones that affect our population and the population worldwide in terms of numbers. 7.2 million people in the United States with Alzheimer's. Over a million with Parkinson's, and about 30,000 with ALS, also known as Lou Gehrig's disease. Now, these disorders are all characterized by neurons dying. In ALS, it's the neurons that make your muscles move. And so they have movement, they have muscle weakness, and eventually even their diaphragm doesn't work. And they have to have a breathing machine to survive. For Parkinson's, the neurons that are dying are in a part of the brain that coordinates your movement. And so they have slow movement and they have tremor, because all the coordination of those groups of neurons that make for smooth movements, etc., are impaired. In Alzheimer's disease, it's mostly in your cortex. Neurons that are involved in memory and cognition and learning and your personality, all those things. And my mother, oh my God, those things changed, let me tell you. So, but the interesting thing, and I'm going to come back to this idea, even though those disorders seem very different in terms of the groups of cells that are affected, the symptomatic course, they do have common features. In all of them, the fundamental mechanisms of cell repair are defective. Cells, like anything else, like your house, like a building, it accrues damage. And if you don't fix that damage, you're in deep trouble. And cells have very sophisticated methods of taking care of damage. In all of these disorders, that repair mechanism is not working. Okay? We've known that for a while. And we have pushed on that to see if there is a common mechanism that we could actually treat all of these disorders. All of them. And I wouldn't be telling you this if I didn't have the data to say, we can. This has been, the last 6 months, has been pinch me, I'm awake time. Because the data we had was so compelling that I couldn't believe it. So I'm really glad I didn't retire and ride my bike across Italy. So I'm on a mission, and we're going to try and push it through to the end and get these, see if these medications work and get them in people as soon as I can. Okay, but I want to go back and tell you a little bit more about the history of these disorders. But please bear in mind that there are, despite the diversity of those disorders and how they show themselves, there are common things. And that's what we're going to go after. Okay. So, let me tell you a little bit about the history of this disorder in particular, since it's so prevalent, such a big problem. First of all, it is a progressive disease. It probably takes 20 years for there to be enough damage of cells to have a clinical demonstration of it. Probably 20 years or so to come to clinical significance. But this relates to the whole idea that this is accrual of damage that the cells cannot overcome. And eventually, you're done. So, what is the typical progression of this disease? Well, diagnosis is typically rather late. So, if you look at this timeline of preclinical, where they don't have any symptoms, but they probably already have biological measurable damage. And they go from that to mild cognitive impairment, where their symptoms may or may not interfere with everyday activities. But when we typically see these patients and they actually get a diagnosis is when they have dementia. And make no mistake about it, at that point, I mean, this is a lethal disease. On average, from the time of diagnosis to death, 4 to 5 years. So this is not just grandpa slowing down and yeah. No. Once they get this diagnosis, there's a very limited window. Okay. So obviously, the earlier we can track the disease, the better, because we might be able to intercede earlier, and earlier is going to be better. So let me back away a little bit and tell you something about the history. We've known about this disease for a long time. It's named after the pathologist Alois Alzheimer, who discovered it in a single patient, 52-year-old woman, in 1906. He wrote a paper about this person saying, oh, this woman had this severe dementia, and I did the pathology on her brain, and this is what I found, okay? And it was sort of a case report. People read it as, oh, this is a very interesting disease, but it's probably rare. So what were the things that Dr. Alzheimer described? 3 primary things that he saw in the brains of these patients. Number 1 was accumulation of something outside of cells, which was called amyloid. It was sort of a throwaway term. It meant an aggregation of something, didn't know exactly what molecules were in there. But it was clearly present in these patients. And it was outside of cells, okay? And those were called amyloid deposits. They're also called neuritic plaques. The second thing that Dr. Alzheimer noticed was that there's something really weird going on inside of neurons. And he used a whole bunch of different stains. So look at this is of course early 1900s, 1800s. You know where all the dyes came from that we use to look at tissues? They came from the fabric industry. Because at that time, production of fabrics and colors and everything was going crazy. It was a huge business. So the pathologist just took all those dyes and said, well, let's put it on samples and see if they help us discriminate different features. And one of those helped them discriminate these tangles of fibers inside of neurons that they called neurofibrillary tangles. We now know that that is microtubules. What do microtubules do? They provide architectural support for the cell, and they also transport stuff along it. It's a highway, okay? We now know that the abnormality in those microtubules is the binding of one particular protein called tau, T-A-U, and it is abnormally phosphorylated, and it binds those microtubules and does a bunch of bad things, okay? But Alzheimer saw this in 1906. And the third thing that Alois Alzheimer noted was there were cells in the brain that showed a lot of lipid, a lot of intracellular lipid, accumulations of lipid that cells are storing inside, okay? And he didn't know what type of cell it was. He knew that it didn't look like a neuron. It turns out to be the 2 other major classes of cells in the brain, something called astrocytes and something called microglia. Microglia are the immune cells of the brain, okay? So those 2 types of cells were accumulating this lipid. And that observation has been largely ignored until recently. Now, so this paper came out in 1906. The next really major step, and oh, by the way, this set of panels down here is just to represent that these accumulations of lipid, they also just occur in aging too. The top panels are a 22-year-old, the bottom are a 67-year-old. So we see them in Alzheimer's patients, but we see them generally accumulate with age too. So lipid is an important part of this biology, at least that would be suggested by the pathology. But the focus of the field has been on that first one, the accumulation of these protein deposits that's outside of cells. That has been the big focus. But I get ahead of myself a little bit. So this was the status of 1906, okay? 70 years later, Robert Katzman, pathologist at the University of California, San Diego, he started looking at many different pathologies from elderly patients, especially those with neurological problems and dementia, and he said, oh my God, this pattern that Alois Alzheimer described in 1906, I see it all over the place. This is not some rare disorder. I see it in a very large proportion of these elderly patients who suffered from cognitive decline. And he wrote an opinion piece to reflect these, what he felt were, and accurately so. This is not a rare disorder, but he found it in many patients. And he basically wrote this editorial saying, we better figure this out because it is a major medical challenge for our society. So 70 years to get to here, okay? Now, there was a lot of investment, and I would say the major idea that came out of probably 40 years of research was this one. And it's called the amyloid hypothesis. And if you read any newspaper article, Newsweek article, whatever, you're going to hear this. And the basic idea was those accumulations of proteins outside of cells, they were the cause of the disorder. If you get rid of them, you're good. That was the logic. And that's not horrible logic. It's okay. Association is not causation, as we all know. So, and there was genetic data that supported this. There are people that have mutations. So this is a cartoon of what happens. You have a protein that normally spans the plasma membrane. So it's got a part of it on the outside of the cell and a part of it on the inside of the cell. And it can be proteolytically cut. In other words, it can be snipped in different places. And the extracellular pieces can misfold, start aggregating, and form this extracellular goo that we call amyloid. And there were two genes that were involved in that processing. And in both cases, there were mutations in people in those two genes that predisposed them to getting Alzheimer's early, and it was inherited. So that was very compelling data that this model was on track, okay? But over time, so those are probably the principal reasons that the pharmaceutical industry has gone after those amyloid aggregates. So the thesis, of course, is if we can remove those aggregates, we will arrest the disease process. That was the hypothesis. Unfortunately, it is only weakly true. So this amyloid hypothesis, which I kid you not, it dominated the field big time, to the point of exclusion of other work, which is the bad side. When you don't understand the disease fully, you better diversify. But this hypothesis has increasingly come into question because it didn't fully explain the pathology, and it excluded other factors that we have come to know contribute to the disease. So it was not broad enough, inclusive enough. And I'll tell you some of the reasons for that. So let me just show you the percentage of people that have detectable amyloid, but they're cognitively fine. Different age groups. Among the 69%, if you just take people off the street and you image if they have any of this amyloid in their brain, 10 to 15% of those people have it, and they're cognitively normal. Among the 70 to 79, higher percentage. Among the 80-plus-year-olds, 30 to 40% of the people who walk around that are of that age group, they have amyloid in their brain, and they're fine. Okay, so clearly there's no direct one-to-one link. Now, if you look at these patients in that 60 to 69 group and you say, okay, what's their fate? How do they do? Well, in 10 years after this time period when they're first evaluated, 30 to 40% of them show mild cognitive impairment. So they get there. And that rate, by the way, is 2 to 3 times higher from people of that same age group that had no amyloid. So it's involved. But it ain't one-to-one, okay? It's not purely, of its own, causative. It may contribute to some degree, but it ain't the whole enchilada, right? So that's just some of the data that's indicated that we can't rely entirely on that as a strategy. So I'm going to jump you up to 2021. First new drug approved by the FDA for treating Alzheimer's disease. It was a drug, it's an antibody drug. It recognizes amyloid. And if you give it to people the levels of amyloid in their brain goes down. It definitely did what it was designed to do. One minor problem. There's no clinical benefit. Now get this. Despite the fact that an expert panel of 12 different scientists said, do not approve this drug, they did it anyway. Oops. Why? Many reasons. Money, hope, that we had nothing for these patients. The reasons for that are complicated. But the bottom line is it didn't work. In the end, even though it got FDA approval, Medicare said, we're not paying for it. $60,000 a year. $60,000 a year, we're not paying for it if there's not good evidence that it works. Thank you very much. So the net result of that, Biogen put up all that money, but no one bought the drug. So by 2024, they quit making it. Okay. The next round were 2 other antibody drugs made by other groups, and these did show some clinical effectiveness. They got rid of amyloid a little bit better. They saw different features of the amyloid aggregates, so they were a bit different. And these 2 molecules, they did the right thing. They published the clinical study. It was done independently. They showed they could measure benefits by these 2 drugs, and they are now both FDA approved. So what kind of effects are they having? Patients show a slower rate of decline. They're still going down. They're just going down slower. On an 18-point scale of cognitive measurement, 27% means they decrease by 0.85 points less. I can tell you, ladies and gentlemen, you wouldn't detect that in your loved one. It's measurable. I believe it. It's modest. Now, on top of that, these drugs, they have some side effects. Intracranial bleeding. It's not good for your cognition. A pretty sizable percentage of patients receive that problem, get that problem. The other adverse side effect is brain swelling, called edema. Also, not good for you. Okay? So those 2 side effects come with it for a pretty modest effect on your cognition. Now, I'm not poo-pooing that positive effect. First drugs that showed any effect, right? This is cause for hope, but it ain't going to solve the problem. So, on top of this, because of those 2 side effects, intracranial bleeding and brain swelling, less than 10% of people that get a diagnosis of Alzheimer's even qualify to get the medication. 8%. So, the effects are modest. There are side effects, and only 8% of the people that receive a diagnosis of Alzheimer's in the clinic from their docs can actually qualify to get the drug. In other words, this ain't going to cut it. So, we need to do better. And you have to understand that there are people in the field that have spent 35, 40 years of their lives on that hypothesis and trying to get those drugs to work. You can imagine how much of a dagger in the heart it is to see those results. But they are what they are. And science is a transgenerational endeavor. We do what we can, we advance it as far as we can, and we hand it over to our brothers and sisters to carry it on. It has been for cancer research, it still is, and it's going to be that way for this disease. It's transgenerational. Okay, so just to probably beat this point down, these drugs, they get rid of the majority of amyloid, 81% amyloid negative with lecanemab. Man, they get rid of that stuff. But they have a modest effect. Okay. So the bottom line is, these antibody drugs that are out there now, they clear amyloid plaques very well, but the clinical benefits are modest. So that kind of takes us up to the present. And I still see things like this in the literature. The amyloid hypothesis versus the tau, it doesn't work this way. It's not one against the other. These are not competitions. We should be thinking diversely and mechanistically. And this, by the way, is something that was in the Alzheimer's Association literature that I found. So my take is that amyloid is certainly involved in the disease, but it's not the sole, nor perhaps the most important element. And it certainly may not be the best target for intervention. Now, changes in tau, they actually correlate better with disease, but thus far, the antibodies directed against tau, no clinical effect yet. Several of them have been in trials in humans. So far, it's been a bust. So that's not encouraging, but they're still trying different antibodies to different parts of the molecule, et cetera. So there's clearly a need for alternatives. So let me tell you about some of the things that suggest alternative pathways. And one of them is that you can't think of these neurons by themselves. They are supported by so many other cells in the brain. And this is just one example. So neurons, of course, they're the ones that transmit information, and they're the ones we want to keep. But they have another class of cell called astrocytes. And one of the main jobs of those astrocytes is to metabolically support the neurons. So literally, what this picture shows you, the astrocyte is in green. It has processes up against the capillary, so it's getting all the goodies from the blood. And then it's metabolizing the glucose down to 3 carbon components, and it's shipping them off to the neuron. It's handing the neuron 3 carbon breakdown products of glucose to support them metabolically, because the neurons, they have a very high metabolic activity, right? They need this stuff. So the astrocytes are central to supporting the health of the neuron. That's not anywhere in that amyloid hypothesis. What is the interplay between astrocytes and neurons in this disease? And we know there are neurodegenerative diseases where the deficit is in the astrocyte. So if you mix astrocytes and neurons, and the astrocyte is diseased and the neuron isn't, the neuron still dies. Okay? So we now have direct evidence that these astrocytes are bloody important. So what are the factors that people have discovered have an impact on this disorder? Well, a lot of them are metabolic. So altered glucose metabolism is very common in this disease. In fact, that observation is more predictive of their outcome than how much amyloid they have or how much tau. Changes in glucose metabolism in the brain. So what are some of the risk factors that we know from studying these populations? Well, type 2 diabetes, big risk factor. Obesity. This is much more prevalent in women than men. We still don't understand that. 2/3 of all the Alzheimer's patients are women, by the way. Why? We don't know. And then there are some various genetic risk factors that are inherited in families, or maybe they're not, they're inherited and they don't say, oh, you absolutely get disease, but they predispose you to it, like ApoE. If any of you have had family members evaluated, you probably know that the patient gets tested for ApoE. ApoE is a gene, the protein product, it handles lipid, central to lipid metabolism. There's one variant called ApoE4 that if you have it and you're homozygous for that variant, both from your mom and dad, you have a 15 to 20-fold higher risk of getting Alzheimer's as you age. If you're heterozygous for it, it's about 7-fold. My mom was ApoE4 homozygous. I'm heterozygous. I don't have as big a risk as she did, so I'm going to try and keep myself zippy, zip, zip, zip as long as I can. So those risk factors, they also scream metabolism and lipid. And remember, Alois Alzheimer's said, hey, there's a lot of lipid accumulation in these other cells. What's going on there? Okay? So we have learned a lot, and we need to pay attention to this, and the beauty is, of course, the community is. So for a moment, I'm going to approach you like biochemistry students. And what I have to do here is relate to you, what is the relationship between carbohydrate metabolism, and lipid metabolism. And simply put, bagels go to fat. Tragically so, for those of us who like bagels. Carbs become fat, literally. Okay, up at the top we got bagels. Converted to glucose, it comes into the cell. And it gets broken down to 3 carbon intermediates first. Okay? And those 3 carbon molecules, pyruvate, they get imported into your mitochondria, which is where you really get energy out of those suckers. The mitochondrion harvests that energy and give you tons of ATP. But what if you're in a state where there's a high energy charge in the cell and it doesn't need all that energy? What happens to that 2 carbons? Guess what? It comes right back out of the cell, and the cell, in 2 carbon blocks, turns it into lipid. And what is a fatty acid? It's a long chain of carbons and hydrogens, and one end, there's a carboxylic acid group. That's it. It's really simple. That's a fatty acid. So those carbons from glucose, if in excess, they become lipid, period. Okay? So glucose metabolism, disruptions of that, and lipid metabolism, they are intrinsically linked. And if you have those risk factors, type 2 diabetes, obesity, you want to take care of it. It will help. Activity helps. Okay, so that's the link between the metabolism of carbohydrates and lipids. So 6 carbons of glucose end up in fatty acids. Here's a study that came out very recently that showed that lipid binding protein ApoE4, it's really important for disease. This came out just in 2024. That really connected lipid abnormalities mediated by that bad variant of ApoE4 to disease. So this is, metabolic contributions to the disorder. Here's another paper. This happened to come out of a lab at Stanford, but one of my colleagues in biochemistry, Melanie McReynolds, was co-author, and one of my other collaborators, Eric Ulian, who's at University of California, San Francisco, was also part of this story. But the title says it all. Restoring hippocampal glucose metabolism rescues cognition across Alzheimer's disease pathologies. And guess what? The deficit was in the astrocytes. They were not able to give them those 3 carbon intermediates. I'm telling you by way of saying, we're making progress mechanistically. So what's the current status of all this stuff? Well, we have 2 anti-amyloid treatments FDA approved in the US. The EU originally blocked them, but has now recently said, okay, they work well enough, we should support their use across the European Union. The United Kingdom National Health Service has still said, nope, they don't do enough, their side effects are too big, we're not going to use them. So they're not universally accepted across the world as good medicines for this disorder. But there's some increasingly interesting data. Like this was a study just published this past summer. People that have ApoE4 homozygous, they at risk. If you put them on a diet, the incidence of them having cognitive impairment drops by 30%. Diet related to metabolism of glucose and lipid in these patients with a risk. And by the way, ApoE4, they can't even get the antibody drugs because guess what? When they get those antibody drugs, they do worse. Okay? So this is pretty big stuff. 30% drop in incidence. Okay? That just came out this summer. Here's another paper, just came out a few days ago. In fact, it's still under review, hasn't been accepted for publication. These guys looked at all the genetic risk diseases for Alzheimer's and said, a lot of these genes, they're not active in the nervous system. They're active in blood vessels. Maybe they're screwing up the boundary conditions between blood and brain. They're also involved in the control of immune system. So there's a lot of complexity there that we still haven't explored, that the literature is now coming out in in a torrent, which is great. Okay. I've got a few minutes more to tell you about what we're up to. That kind of is my summary of the world of this disorder as I see it at the present. So I'm going to tell you a little bit about what we've been doing, and it relates, of course, to some of these things I've been describing. And I've had help from a grant from our College of Science called Lab Bench to Commercialization, another grant from the Penn State, from the VP for Research's office has helped us, and I've had help from a venture capital, Pennsylvania-based, and from a company called Selleck. So this has been a great relationship, and that's what we're here for. It focuses, why are we doing this? So let me just give you a little bit of a taste of what we've been doing. So this is a slide I showed you before, where I indicated that there are common and early pathologies. And that has been the focus of our work. So, by the way, lipids are degraded and harvested for energy, guess where? In the mitochondria. So if you have mitochondria that aren't working well, you ain't degrading that lipid, okay? And by the way, mitochondria, they have a very limited lifespan in our cells because they're doing some very dangerous chemistry where literally electrons are being transferred. Individual electrons being passed on, transfer from one protein to another. And the way this works is that transfer actually generates a proton gradient, like a little battery, and that proton gradient is what drives ATP synthesis. So, but those little electrons, those buggers get away from you, and they do damage. Like any machine, it's going to fail. So if you don't get rid of the damaged mitochondria on a regular basis, you're in deep trouble. So it's a two-edged sword. These mitochondria, man, they give us tons of energy. They're incredibly efficient, but you have to watch them. So when they accrue enough damage, you got to pull them and make a new one. And that's what cells do. But if you don't have the cell repair processes to do that, those bad mitochondria expand. And then they can't degrade lipid because they're not working right. The point I'm trying to make is they're all connected. Cell repair, mitochondrial abnormalities, lipid metabolism, glucose metabolism, they're all connected. This path that I've been on that Dan described for the last 9 years, it really started with this student, Claire Reynolds Peterson. This is her at graduation. And she was the one who made these observations that I could not let go of. And what she showed was that a class of molecules that we had been working on for a long time, they regulated cell repair. And their normal function was to actually inhibit cell repair. So when we compromised what they did, we pulled away that inhibition and cell repair went up. And that was the observation she made. And she said, that could be useful. If we can boost cell repair broadly, maybe we can have these cells recover from whatever damage they may be accruing. That was the observation she made. That was her first paper in 2017. I also want to say something about, the power of undergraduates. So this was my class from my lab. They all worked in my lab at least 20 hours a week as undergrads. I mean, on top of their other responsibilities, I would say if you're going to come into the lab, you got to be part of it. This is not a game. This is not some little training exercise. You are going to do experiments. You are going to be responsible for those experiments. You are going to contribute. If you want to do that, you're in. If you don't, okay. So it's a big commitment on their part. This was a set of 6 that graduated in 2023. Mackenzie on the left is now in physician assistant school at Temple. Alyssa Connell is an MD PhD student at UMass. So she's getting a total free ride. Free tuition for medical school, free tuition for graduate school, a $40,000 a year stipend while she's a student. She will walk away from the world's best education and she will not owe a dime, which is a good thing because she didn't have a dime, and nor did her family, okay? So Alyssa and Shalini is in charge of clinical research protocols at City of Hope in California. Alex is the tall guy, good baseball player. He's in medical school at the Geisinger Medical School. And the other two gentlemen are at Virginia Medical School and Ohio State. So I tell you this by way of saying this is what happens when students are given opportunity. It affects the trajectory of their life. It affects their perception of their own abilities to make a contribution. And they make my life better and richer for knowing them. So I now have 9 undergraduates in my lab. It's kind of painful in the sense that you train them and you have to watch them leave. You know, and you value them as colleagues because their skill sets get better and they're more productive, and you like them and want them there, and they become your friends as well as your students. But, you know, this is the way it goes. It's just like having children. You do the best you can for them, and then they go on to hopefully bigger and better things. So this was the group from a few years ago, and this is Alyssa when she got admitted and took her white coat ceremony. And I put this out because those are the 4 MD PhD students that were accepted into the program in that year. They only accepted 4. So this is the kind of competitiveness that our students are perfectly capable of doing. So that was a good day. I drove up to Worcester to participate in her white coat ceremony. Okay. I'm going to close out with a little bit about the logic of our strategy and what we're trying to do. Cell repair is inherently a breakdown process. You have a damaged mitochondrion, you want to recycle all its parts. You want to degrade it, take its parts, reuse them. Protein aggregates, they're misfolding. You want to degrade them into the amino acid building blocks and reuse them. So it's inherently, in biochemical terms, a catabolic process. It means we're breaking stuff down. The opposite is anabolic. When we're growing, we're building stuff. Cells getting bigger, more ribosomes. We're getting ready to divide if everything's really good. So repair is an inherently catabolic process. It removes damaged mitochondria, it gets rid of aggregated proteins, and it helps mobilize the lipid. And all of those are defective in all of those diseases. The counter is that signals between cells that are, say, grow. Insulin is one of those. Insulin-like growth factor, related one. Fibroblast growth factor, epidermal growth factor, platelet-derived growth factor. All these are protein molecules that are secreted by one cell to talk to another, and they say, grow. Make more machinery. Don't degrade anything. Because if you're going to grow, you don't want to be degrading stuff. So these growth-promoting signals are inherently inhibitory to cell repair. So we've taken advantage of this relationship, and we found a way to block that inhibition. So put in another way, we are moderately inhibiting growth signals to boost the level of repair. So here's a cartoon that makes the point. We have these growth factor and nutrient signals that are saying grow, 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 and those signals inhibit these repair systems. So they inherently inhibit it. So if we dial back on the growth, we can raise the repair. Okay, that's the logic. And I wouldn't be telling you if it didn't work. So this is overall how you go about trying to develop a drug. So the major work in this business is showing that you have a good target for the disease. If I affect this particular gene or its protein, does it do something good? Does it suppress disease? And if that works, then you say, oh, that's great. And we do this genetically. And we use models in mice and in stem cell-derived models, all kinds of different models for disease. That's most of the work, is proving that you have a target that's a good one. And then you have to get a molecule that affects that target. So this first part, identify a single gene to target, that takes most of the work. And then you have to figure out, can I find a small molecule, that, i.e. something that can be turned into a drug that inhibits my target, and can I prove that it 2 works in suppressing disease, and then you can go to a drug development program. And that red arrow says where we are in our project right now. Okay, and we have many different models, and we've used them. We have, believe it or not, we have fruit fly models of these disorders. We design that model to reflect the human disease, okay? And you can use that both to assess, is my target a good one? And you can also use it to see, do I have a small molecule that does the job? I just want to show you one experiment that we did in our lab where we're expressing a gene involved in Lou Gehrig's disease, ALS. So we've taken the human disease gene and we've put it in a fruit fly, and we're only going to express it in the retina. And we're going to see if it kills neurons. And it does. So this is a normal, happy fruit fly with a retina. It's fully intact. That's what happens if you put that human disease gene in it. So that black is the scarring, because underneath that cuticle, remember, insects have their skeleton on the outside. Underneath that are all those neurons dying. So you have this beautiful readout of the disease. And it's caused by the human disease gene. And when we do that, and we inhibit our gene, we convert this to that. Okay? So that shows that our strategy is working. So this is a genetic experiment. So we've developed small molecules that inhibit the protein product of this gene, and we're using all these models to evaluate their activity. And the bottom line is, they work. Let me just give you, forgive me, I have to show you this one. So this is a Parkinson's model. And there's a human gene called PARK2, and if you have it, if you're homozygous for mutation in that gene, your mitochondria do not get identified and degraded when they're defective. It's a surveillance gene, okay? It's called PARK2 in humans, and there's a very close relative in fruit flies that we've called Parkin. Now, what are the cells that are really in trouble in the fruit fly when they don't have this gene that's involved in getting rid of damaged mitochondria? There are cells in the brain that die, but there's another cell type that really needs mitochondria. It's the flight muscle. So that big thing right there in that upper micrograph, that's the flight muscle. That has to contract 200 cycles a second to get that little bugger off the ground. Okay? Energy demand, lots of mitochondria. And guess what? When they have a mutation in that Parkin gene, those mitochondria do not do well. And the muscles degenerate, as do neurons in that same model. But it's a lot easier to look at it in a flight muscle. So let me show you what our gene target does against fixing that. So there's a normal fruit fly flight muscle, and all those green things, it's just mitochondria. We've tagged only the mitochondria. That's a happy and healthy muscle. This is a Parkin mutant. Not so good. And this is what happens when we reduce the function of our gene by only about 20%. 20% going down on our gene, and those muscles are almost indistinguishable from wild type. This was the result that 7 or 8 years ago convinced me we should do this, because it showed that moderate changes in our target have big impact. And they didn't do anything bad. That's what you want with a drug. Something that you don't have to clobber the target 100%. You can clobber the target by 15, 20%, so you don't have other problems that might come with higher concentrations. You can inhibit a single target and suppress deficits in every major neurodegenerative disease. Here's the current gang. So I had wonderful people to work with. These are some of the undergraduates that are in the lab now that have done some of the work I've described. The top right hand is Costanza from the University of Bologna Medical School, who was out here for a while. And in addition to this team, this group on the left, Corinne and Brianna, they did a lot of the ALS model work. Megan, to my left, in that picture, was the one who did the iPSC model in San Francisco, and Anusha joined the lab about 8 months ago, and she's working on a number of things. So the other point I want to make is you can't do this as a solo lab. The biochemistry on these molecules that I haven't told you anything about, that's being done by my collaborator at University of Georgia. The mouse model we're working at University of Alabama. The stem cell base, that is, cells taken from patients with the disease, converted to stem cells, differentiated to neurons or astrocytes. That's at UCSF in California. So my student Megan was the one who went out there for a couple months and did that work. So anyway, we are making progress. We're hopeful. We have a long ways to go, but we're hopeful. But it's not done without expertise and talent and contributions from all these other people and all these other labs. So this is the last one. I want to thank you for being here. Thank you for listening. So this little QR code thingy is if you want to make a contribution. And there are many ways you can do it, including your time, by the way. I encourage you to get involved with your college in any way you wish. That little QR code will take you, and there are many different ways that you can help. So the first one is helping my lab directly. The second one, I love the second one. The second one is providing money to pay our undergraduates to participate in the research. It's like a mini scholarship for our undergraduates as they work in people's labs like they have in mine. And the third one is also important. This is a discretionary fund that the dean has, where she can use money strategically on things that are important for the college, because you can apply it where it's needed. So our dean, this is a good thing. It gives her, Tracy Langkilde, our current dean, it gives her means of saying, oh, this project over here, this young scientist needs help. And sometimes small amounts of money at critical times in science can really make a difference. So that is another way. But anyway, that's just if you're interested and want to. Any way you want to contribute to our college, your engagement, your thoughts, your interest, it all matters. And with that, I will close, and thank you. I'm happy to answer questions.
The impact of GLP-1s in slowing the disease for you to be able to get this through fruition, what are you thinking about with that?
So there was a very big clinical trial just recently on GLP-1s, tested against patients with mild cognitive impairment. Didn't do anything. Interestingly, the studies that had preceded that one were retrospective, right, looking back. And the patient group was very different. The patient group were people with type 2 diabetes. And in that group of patients, and they were also assessing how many showed incidents of cognitive impairment. And in those, and they were big studies, 1 million people. Those studies showed a 30% reduction in new incidents of Alzheimer's among those type 2 diabetics that got treatment. The current study was not type 2 diabetic patients. In fact, they were excluded. The new study that showed no effect were people that had mild cognitive impairment already. And they also used the orally available drug instead of the injectable one, which they just don't know as much about it clinically. So I think GLP-1s, do they work for type 2 diabetes? You betcha. Do they work for reducing weight? You betcha. Are those contributors to Alzheimer's? Uh-huh. So my suspicion is that if you look at people with cognitive impairment already, they're not going to be as effective. And for people that have type 2 diabetes, et cetera, the retrospective data certainly shows fewer people march to cognitive decline.
Well, first of all, thank you so much for your work, and this approach to cell repair sounds so exciting. My question is, in the August issue of Nature, there was a article about a Harvard study that showed that very small amounts of lithium orotate could decrease Alzheimer's and even reverse it. Now, they only did it in mice. They were like where you were. They haven't had human clinical trials yet. But what are your thoughts on that?
So it's a very interesting study.
Lithium, one of its effects is to regulate a very important signaling pathway called Wnt, W-N-T. So we know it as a bipolar treatment. It's used to treat people with bipolar disorder. But it has metabolic effects too. So I think it's a very useful and important area of inquiry, is what I think.
Thank you for a great presentation and continued success. Estrogen. If Alzheimer's is affecting postmenopausal women, will estrogen play a role in helping that situation?
The relationship with postmenopausal state definitely matters. The problem, what we don't yet know is it as simple as changes in estrogen levels. Estrogen replacement therapy does not seem to correct it. That doesn't mean that, menopause is a very complicated set of events. So this is one of those areas that deserves a lot more research. It doesn't look to be simple. Estrogen replacement therapy will save the day, is the short answer to your question. But what else may be going on? It's a very active area. So I think it's clearly related to the events that occur with changes in those hormone levels. Exactly how they're tied together, we don't understand yet. But it deserves a lot more work since 2/3 of our patients with Alzheimer's are women.
We have a question from online. Any role of vitamin D in these metabolic abnormalities?
Well, vitamin D is very important for repair. I don't know of any data specifically related to neurodegenerative disease, but that's not my area of expertise. It may be out there, but certainly repair mechanisms and vitamin D plays a role in that. It may well. I mean, the good news, ladies and gentlemen, is that since the amyloid hypothesis has clearly shown it's not going to achieve the end we need, the good news is there's a flowering of diversity of science, like the lithium study or dietary influences on ApoE patients. The good news is that the field is in a dramatic period of growth and diversity, and this is a great thing. And let me tell you, that's had an influence. But yes, we have a lot of diversity of strategies that are all worth delving into.
What do you anticipate the timeline might be until you have a clinical trial? 2 years, 5 years?
It is possible that our strategy could be in phase 1 clinical trials in humans in 3 years. Frankly, it depends on how much money this company that we're engaging with decides to drop into it. Money is speed. So if they put in enough money, we might be able to have phase 1 safety trials on these molecules in 2 to 3 years. I'm hoping it'll be on the market in 6. That's a bit optimistic. The company that we're engaging with, they are a great group of people to invest. And what I'm trying to do is give them every good reason to do so. And they have been very responsive and very helpful. And I've tried to enlist them in this is not just a job. This is a mission. You got to believe, because there are going to be challenges along the way. It's not just about money. It's about, making sure that we give people 10 to 15 more years of cognitive life. And that group feels that way. So I'm fortunate to be associated with them. But if Bill Gates called me up and said he was going to sell his yacht and give me the proceeds, I would not argue against it.
So Scott, we're gonna go through a couple more questions that are online.
Will drugs that block IGF-1 have beneficial effects on neurodegeneration?
I don't know of specific data to that point, but IGF-1 is a growth factor. I haven't told you the molecules we're after because that's part of our patent, but I can tell you that they engage and promote the signaling of many different growth factors, including insulin-like growth factor, et cetera. And when we knock down that signaling, things get better.
Are you allowed to say exactly what protein your small molecules are targeting?
You give me 20 million, I'll tell you what it is. We are in the midst of filing provisional patents. We're in the midst of engaging companies to support it. And one thing is just a truth of this business. if there's not money to be made, the best science in the world will not get developed. So we have to be attentive to the finances, because without it, it's going to take 70 million to get this to safety trials in humans, okay? Someone's got to be willing to do that and make the money back, right?
What are some of the side effects that you can anticipate from your inhibitor molecules,
Oh, good question.
specifically growth pathways?
Very good question. That is the $50 billion question, because you need to affect your pathway sufficiently to do good and do no harm. And you know something, we know that we can do that in a mouse. We know we can do it in a fruit fly. We know that we can moderately affect our pathway with no deleterious effects and do a lot of good. But until you put it in a human being, you don't know. You just don't know. You have to put 1,500 people on placebo and 1,500 people on your molecule, and you see what happens. And only then can you say, we're in. So the biology of humans is different, and the biology of a small molecule inhibitor, that's another level of complexity. So there's a lot to do still, but you got to give it a shot.